Good afternoon from NASA's Kennedy Space Center in Florida, and welcome to today's news conference. SpaceX is due to launch its Dragon spacecraft tomorrow morning at 10:10 a.m. Eastern Time. There's quite a bit of supplies for the crew of Expedition 34, as well as a significant amount of science on this mission, both going to the space station and coming back home at the conclusion of the flight. Here to give us more details about what's ahead is Julie Robinson, the International Space Station Program Scientist. We're also joined by Simon Gilroy, researcher and scientist from the University of Wisconsin. We're also joined by Marshall Porterfield, the Life and Physical Sciences Division Director up at NASA headquarters, as well as Mike Johnson, Chief Technology Officer from NanoRacks, as well as Mike Roberts, Staff Scientist for the Center for the Advancement of Science in Space, or CASIS. We're going to hear from each one of them. We'll get started with Julie.
Thanks. SpaceX 2 is a really important mission for us because the cargo that goes to and from the space station is key to completing a research mission. There'll be about 330 kilograms of supplies for research and experiments that go up on the flight and about 571 kilograms that come down. And it covers all the broad disciplines of research that we do on the space station, including human research, biology and biotechnology, physical sciences, and education activities. And all of these supplies go to support the over 200 investigations that are active on the space station today. Some highlights of some of those experiments are some new cell biology hardware and some new hardware for doing genetic analysis on orbit. And those really help step up some of our life sciences capabilities. And later today, you'll hear both from Dr. Gilroy talking about his specific experiment and also from Dr. Porterfield to talk about the overall ramp up in the life sciences program at NASA. We also have a flow cytometer that's going to provide the ability to count cells at a very fine scale. And in the area of physical sciences, we have commercial studies by organizations such as Procter & Gamble aimed at making products, improving products by studying colloids, and studies of spacecraft materials and other important areas for NASA's exploration mission. So you'll be able to follow the progress both of the launch of these samples, and then as the samples come out and are used on orbit and the experiments are completed, you can follow all of that through our social media feed, which is at ISS_research. With that, I'll let Simon Gilroy tell you about some of the experiments that he's going to be doing.
Well, thank you, Julie. So this is a very exciting opportunity for us. We're part of a package called Brick 17, which is to put plant biology into space. And there are a couple of different questions which we're trying to answer between a couple of groups working there. We're all using this experimental material. So this is Arabidopsis thaliana, or mouse-eared cress. We always like to say this is the lab rat of plant biology. It's the plants that we understand the best of. And so we're putting these into space to begin to pull apart a couple of biological questions using the ISS as what it is, which is a fantastic laboratory doing experiments that we simply can't do on Earth. So particularly for the experiment we're interested in is related to the fact that in space, conditions are different from what they are on the Earth, and some of the challenges that biology faces are very intuitive to us. So, for instance, astronauts losing muscle mass and bone mass makes a lot of sense because of that weightless environment. But there are other challenges up there for biology which are a little bit less intuitive but equally important. And one of those is that gases move differently in a weightless environment from the way they do on Earth. And one way to think about that is a hot air balloon rises because the hot air weighs less, and so you have buoyancy driven movements, and that doesn't occur in space. So that movement of gases driven by convection does not occur in weightlessness. And so if you think about what that means for biology, imagine that you're a plant and you are breathing, you're respiring, using up oxygen. That mixing of gases doesn't occur, and eventually you will use up the oxygen nearby you. And so you can suffocate a plant under those conditions exactly the way that a human being could suffocate with lack of oxygen. So our experiment is that we've engineered some of these little Arabidopsis plants. We've changed their signaling system that they use on Earth to sense oxygen conditions, and we've made that more sensitive. So one way to think about that as well is that those plants on Earth, if they're challenged with low oxygen, so when would that occur? Imagine you've flooded a field. The root system in that flooded field doesn't have oxygen available to it, and it sends out a lot of stress signals. we've turned up the volume on those stress signals. Now, in space, we think there's a similar kind of issue going on with low availability of oxygen, and so we will have engineered these plants, we hope, to be able to deal with that stress a lot better. And so the experiment that we're doing is sending Arabidopsis plants into space, and then we'll bring them back down and ask, did they perform better in space? We'll look at things like how big the plants are, what genes they switch on, and all those kinds of things.
Okay.
I'm really happy to be here with Simon today in supporting his research. I recently came to my post at headquarters from Purdue University, where I'm a faculty member. So I've lived the life of a NASA PI, and it's really a privilege for me now to be here and support the community and support world-leading researchers like Simon Gilroy. The other reason I'm happy to be here, because Simon's work that he's described to you was also the topic of my PhD dissertation, so I'm kind of interested in how this turns out. So I guess I wanted to bring up or talk about some of the reasons why we do work with plants in space, and why we do research with plants in space. One is that the stress responses that Simon is talking about also occur on the ground. It is an important agricultural problem. So the more that we learn about that, we potentially can take that knowledge and serve the agricultural community and solve problems that are real agricultural problems here on the Earth. The other is that plants also act as sentinels to some extent. Many of the mechanisms for how plant cells grow, divide, respond to stress are conserved across all living organisms. So we may be learning things about plants that apply to human research, for example. The other is that this work really does support the development of long-duration human exploration systems in the future, in that possibly one day we may be developing space missions that rely on bioregenerative capabilities. So plants in such a system like that would not only provide food for astronauts, for crew over long periods of time, but also potentially could be involved with recycling the atmosphere and water purification processes. So this is an example of the type of research that we are doing today on the ISS that has impact in the future in terms of exploration. So we do see the opportunity right now with the space station as being very important to do the relevant research in biological systems and in human research that solves some of the problems that Simon mentioned earlier, bone and muscle wasting, also immune responses that are critical to understand to protect astronaut health over long periods of time, over long missions in deep space exploration. So one of the things we're starting to do right now, and we're going to be rolling this out over the next few months or so, and I'm working closely with Julie Robinson in the ISS program office, develop new platforms to enable more research by researchers like Simon and allow more people to have access to ISS research capabilities. So we're developing more open source innovation approaches for doing science. We actually refer to it as open source science, where we'll be doing large scale experiments using the most advanced analytical tools that are available today to create bioinformatics database-based research opportunities for researchers on the ground to be able to study the responses that are measured in space. And these open source approaches will be funded by NASA, but at the same time they'll be open for anyone. Even I envision high school kids doing their science fair projects based on results that they've mined using bioinformatics tools from these databases. Thank you.
Okay, I'm Mike Johnson from NanoRacks, and we are extremely excited about the SpaceX 2 mission. We have 2 sets of payloads, actually four modules about this size here on the vehicle. And the first module that we're flying is a commercial protein crystal growth experiment. we're very excited about this. We've been working in conjunction with Emerald Biosystems and CASIS, and this experiment takes advantage of modern advances in microfluidics. And on this one flight, we have about 25 proteins that we're flying. Each protein has 400 different sample conditions, so a total of 10,000 different sample conditions. It all fits into about 4 cubes about this size here. Now, what does that mean to, say, commercial protein crystallography? Well, for the first time in the history of this rather long protein crystal growth program that has occurred over the years at NASA, we're going to get long-term exposure on the space station. In addition, we're going to be able to perform matrix, basically you could say using Edisonian methods of trying every single possible combination of protein concentration to figure out how these protein crystals will grow in microgravity. And we're very excited about this because in the past, with this new state-of-the-art hardware, in fact, this hardware, it was developed and is commercially sold by Emerald Biosystems in the Boston-Seattle area, has become basically a de facto method of growing protein crystals terrestrially. And for the first time, we're going to get the opportunity to fly this in microgravity and try it in space. We have a lot of researchers very excited about this because we're able to remove one variable, and that variable is gravity. And especially on the space station, we have a fantastic situation. I might also add that on the SpaceX 2 launch vehicle, the Dragon capsule, we're using the glacier freezer. The glacier freezer is fantastic for us from a commercial standpoint. It's solving a lot of problems. We load our little crystal plates. Each of the plates, we have 25 plates in our sample set. They're about the size of a microscope slide. And each one of these plates holds, as I said, about 400 different sample conditions. And what we do is we load these on the ground, freeze them, and then fly them up frozen in the glacier freezer. Once they get on orbit, they'll thaw out, and the crystals will start to grow. And we'll examine these crystals on orbit with a small microscope that NanoRacks has in its facility. So we're very excited about this particular project. And the second set of experiments that we're flying is a set of experiments developed by a series of high school students. In particular, Valley Christian High School in the Silicon Valley area. It's a fantastic organization. They've developed a set of experiments that fit into a semi-standard cube. Basically, 4 experiments fit into each one of these main modules, and the module itself connects with NanoRacks frame on orbit through a USB port. As Marshall was talking about earlier, at NanoRacks we've concentrated highly on using standardized interfaces such as USB connectors. As you may know, there are something on the order of 6 billion USB devices in the world. I think that's about 1 USB device per person. And in fact, I see a whole bunch in this room right now. So the folks at Valley Christian, in conjunction with 9 other high schools and organizations, have put together a series of 12 different experiments. In particular, I'd like to call out they're bringing in schools and institutions from all over the country. The Hawaiian Girl Scouts are flying a lettuce growing experiment. That'll be very interesting. We also have some folks from Minnesota, Minnehaha Christian High School, or I'm sorry, Minnehaha Christian Academy. They're working on an experiment to see how paint adheres in orbit and how it dries in orbit. That's something we haven't really done before. There's also an electroplating experiment out of the Valley Christian High School team that I think this is I believe the second time this is flown, but this is pioneering area. So I don't think anyone on this team is over 18 years old except for the mentors. And all these kids have done a fantastic job. So as Marshall said, we're starting to implement high school projects on orbit. To me, it's fascinating. I've been in this business for about 22 years, and the unprecedented access that the International Space Station is providing, and also the transportation systems, in particular SpaceX Dragon, that they're providing to the research community is phenomenal. It's a good time to be in space right now. And yes, with that, I'd just like to thank all of the help we're getting from the NASA folks to make this commercial program a success, and also CASIS. As I said, it's just a great time to be in space.
My presence here today is to reintroduce you, or introduce you for the first time if you haven't heard of CASIS. CASIS is the Center for the Advancement of Science and Space. We are a non-profit, non-governmental organization that was created with the intent of defining new research pathways to utilize station. So our service to NASA is to increase the ability of scientists to get their science and technology development up to station. As a non-governmental, not-for-profit organization, CASIS has the opportunity to engage in the marketplace in ways that government organizations and NASA couldn't. One of those is that we interact very closely and work very closely with implementation partners like NanoRacks to enable them to find funding for their hardware and to identify scientists who have questions with Earth benefits that can benefit from the unique environment of the space station. We're very excited about the SpaceX 2 launch because it represents a pivot point in CASIS's relationship with NASA, in that as we move forward from this point on, more of the science that is going to go to ISS National Lab that has direct Earth benefits is going to be managed by CASIS and its organization. So we view this as a door opening, a new window opening, and it is truly an exciting time for scientists in space, because we truly are going to be able to do a lot more science on station from this point forward because of the ability to engage commercial, academic, and other institutions who previously didn't consider ISS National Lab as a viable platform for their science. To give a bit of a plug, we have some of the first science that will be going up to station that is going to be managed from cradle to cradle by CASIS will be going up later this year for increments 37 and 38. We're very excited to announce that that mission for CASIS is going to be known as the Advancing Research Knowledge, or ARC-1. It symbolizes, again, this transition to ISS National Lab as a national lab entity, where a number of investigators can have the opportunity to get their science up there, whether they're coming from academic institutions or from commercial interests.
Okay. We're going to take some questions now from here, and we have quite a number of the social media people that are here as part of the NASA Social. We'll start with Jay over here on the right-hand side. Oh, we'll start with Ken. Okay.
Jay's behind me.
No, I know.
That's what I thought you were pointing to. I'm sorry. Ken Kramer for Spaceflight Magazine, Universe Today. For Simon, I have some questions for you. If you could describe your experiment in a little bit more detail. What did you change in these plans? Is this the first time these plans have been altered? And what would be what's the effect on Earth? So what's the control do these changes have on their survival on Earth? Thanks.
Excellent questions. So as part of a bigger research program, we've been trying to understand what happens when plants get flooded as an element of an agricultural problem, but also it's a very fundamental problem about thinking about how biology, like a plant, can tell what the oxygen levels around it are and then deal with that. So there are cellular signals which are generated almost instantaneously when the oxygen levels in the environment drop. And we are at the forefront of science here, so we don't know how that sensor works, but we know what the signals are. And so one of the things that changes is a signature of calcium ions within the cytoplasm, and that's a classic cellular regulator. So we know a lot about the dynamics of that, and we also know a lot about the genes which are involved in making that signal work. So taking that information, which is we've drawn from a lot of other people's research and our own research, and pulling all of that together, we were able to target a couple of genes which we are fairly confident are key node points, key pivot points in that signaling cassette. And because of this little plant here, because of Arabidopsis, the plant biologists have a fantastically powerful set of tools to be able to go in, pick a gene, and manipulate that gene. And so what we did was we took the identity of those key genes, signaling genes, and we went in and we knocked them out. And it turns out that those genes are the off switch for the signal. And so we take the off switch away, and so when oxygen levels drop, this calcium signal is generated, and it's much, much bigger than it would be normally on Earth. And that triggers a much larger kind of what you might think of as an oxygen defense response in the plants. And that's why on Earth, we know that these plants are resistant to low oxygen stress. So in space, our hypothesis is that the same kind of thing is going to happen. And so the low oxygen stress that the plants see in space, these plants should just behave a little bit better under those circumstances. On Earth, the original reason for beginning to think about this, it's one of those happy coincidences or coalescence of research programs where there's a problem which is a space problem, but that space problem is an Earth problem as well. And so plants having to deal with flooded fields, plants encased in ice, you know, it doesn't happen in Florida very often, but we're from Wisconsin at the moment. It is cold up there. And there was actually this white stuff on the ground. And so plants which are encased in ice are locked away from a supply of oxygen. So there are many naturally occurring environments where that stress occurs, which is why plants can deal with it in the first place. And what we're doing is tinkering with that system, and it just turns out that the space station is the perfect place for us to do an experiment where we can set up this low oxygen environment in a way that is simply impossible to do on Earth. So it's taking that ground-based research, using that information in space, but the information we're going to get back from space is going to plow straight back into that research program, which one of the goals of it is to understand, you know, when a farmer's field is flooded, what's going on, and the long-term goal there would be to be able to do some breeding or something to make crops more resistant. So it's that nice circle of research.
Okay, over here on the left, then we'll come to Irene next. Go ahead.
Hi, I'm Miriam Kramer with Space.com, and I have a couple questions. One is, have you heard of anything fun being sent up in the capsule, either for the crew members, or are there any other experiments that you'd like to highlight? Anything at all like that?
Well, we could, you know, it's difficult in this amount of time to highlight. There are roughly 43 investigations with something either coming up or coming down on this flight, so it's definitely hard to pick a favorite. I think one that's particularly interesting is a combustion study that we'll be doing where we're burning actual spaceflight materials. And through the course of earlier research on the space station, we've learned that spacecraft materials burn a lot differently in space than we thought. So we set up a whole spacecraft fire safety program based on how things burn on the ground. Now we're finding that the soots are much larger, that things burn differently, and that's really transforming the way that we plan for space missions for fire safety in the future. And so that's an example, something very different than anything we've talked about up here.
Okay, Irene, then we'll come over here.
Thanks. Irene Klotz with Reuters. Hi, Julie. You mentioned Procter & Gamble in your opening comments. Could you elaborate a little bit on what their involvement is and if there's any other large companies that are backing any research?
Yeah, so Procter & Gamble's been a collaborator of academic researchers at Harvard University and Cornell and several other universities that have done colloids research on the space station for quite some time. And in this particular case, in the study we call ACE-M1, those samples have been particularly tuned to some fundamental questions about how colloids behave that you can only study in space. So remember, colloid is something like a paint. It's a mixture of a liquid and a solid. And colloids are paints, laundry detergent, all kinds of consumer products, fabric softener, all of those things are colloids. And optimizing the processes of separation and the process of mixing for those is an important industrial research area. So the fundamental research research about colloids that's helping us understand things like the critical point that's being done at Harvard has fed into this Procter & Gamble collaboration. And so what they'll be doing is using samples that are tuned to answer some of their specific questions about the physics of the systems that they have in their products. And they'll be using the light microscopy module on ISS and actually looking at the separation and the mixing and the processes at the molecular level, or at the particle level at least, in these mixtures of colloids. There are a number of other companies, and why don't you go ahead and take that one, Mike?
One of the experiments in our in the Valley Christian set is sponsored by Valspar paint, and we're also looking at colloids in a microgravity situation, how paints would be used in microgravity. It's something that hasn't been done. They haven't had to paint the space station yet, I don't think so.
Okay, Jay.
Jay Barbary with NBC. Julie, a two-part question. You're talking about the science going up on CRS-2. The science you're bringing back, will that be all science that has been on the station and returning to Earth, or are you mixing apples going up and back? And part two, when we were building the space station, we were constantly criticized that all the work going on up there was construction, engineering, development, and nothing was going to science. Now that ISS is completed, what percentage of the workload for the crew would you estimate goes for science each day, and how much science are we really doing on the International Space Station?
Sure. First of all, we have some experiments that are round-tripping. If they're short enough duration, we can take them up on Dragon, put them on the space station, do a little bit of operations, and then bring them right back home. And some of those are a demonstration of the new cell biology hardware that we're flying, the Brick 17, so Simon's work is in that category. Also, a stem cells investigation that's being flown by our Japanese colleagues, and an experiment on materials coarsening. So when you take an alloy and you solidify it, it coarsens over time, and an experiment on that. Those are some of the ones that are round-tripping. Then we have a number of samples that are coming home that have been built up over time on the ISS, and some examples of those are all of the human research samples, things like blood samples, urine samples, other kinds of things that accompany our human research program. We also have hardware that we've finished using on orbit that will refurb on the ground and then provide the opportunity for the researchers to launch again. Some of those things are in the physical sciences as well. So that's just some examples. And then to answer your second question, you know, the boundary between the assembly period and the post-assembly period, where research is the primary mission, is just, it's an extraordinary thing to have experienced. Because, you know, I was program scientist starting in 2006, 2007, and so I worked through that entire transition phase. And when I first started, we would have 7, 8 experiments max sometimes, or maybe only 5 or 6 hours a week. Our strategic goal after assembly was done was to get to 70 hours a week total across the whole space station, 35 in the US operating segment and 35 in the Russian segment. But what we're seeing happen now is we're pushing that boundary as we get more efficient with our operations. And so just a few weeks ago, we had the record-setting number of hours that went to US research. It was 71 hours in a single week went to US research. And so what that translates into is we see the throughput going up, we see scientists having repeat access, we've got SpaceX flying now, and all of a sudden the laboratory's really achieving that original potential. It just, you know, it took a while to build it, but now that it's built, it's an amazing platform.
Okay, Marcia.
Marcia and Associated Press. I'm just wondering, is it just the one type of plants that are going up, or are there more different kinds? And perhaps what quantity of plants? And I see the Medaka fish are coming back, or any creatures going up, as small as they might be.
I think we can see for Brick 17, there are 2 sets of experiments which are going up. There's ours, which is seedlings of mouse-eared cress, and there's a group from the University of Florida who's putting up cell cultures. So that's taking parts of plants and then turning them into individual dividing cells, and to ask what the difference is between how they respond and how an intact plant responds. I think I'd have to ask Julie about the other plant material.
We have some other plants coming home, plants from the plant signaling study, which was a different study that was in our centrifuge, our EMCS centrifuge, that was looking at how plants know which way to grow down and which way to grow up. Those are also, model plant material, but it's a different investigation coming home. We do have the samples from the Medaka osteoclast study. That's the study where Japanese researchers are looking at the cells that form fish scales, which are osteoclasts, just like the cells in our body. The cells that form fish scales are very much like the cells that remodel bone in our own bodies, and so it's a model system for looking at the way those cells function. And their samples, which are, preserved samples, are coming home on this flight.
And on the NanoRacks payloads, we have actually 4 different plants that are flying, or I should say in the seed state. They're going to grow on orbit. And we have at least 2 different bacteria experiments, so we have little critters.
Okay, over here on the left, then we'll come over here.
Hi, I'm Anna Micah with Social Media, and my question is for Dr. Gilroy. I also graduated from UW, so I understand the cold weather. But it has to do with the plants that you're studying. Are they the same plants that they studied not too long ago on the ISS, that they did the challenge for kids where they could grow the plant seeds in the classrooms? I don't know if you're familiar with that. Is that something that you're looking at perhaps doing? I'm also a teacher, so are you going to bring those seeds back so kids can grow them in the classrooms so that they can have that connection?
I don't know about that. Yeah.
We've grown a couple of different kinds of seeds in different educational activities. We have grown Arabidopsis. We've also grown basil seeds. We've also flown basil seeds that were brought home, and then we've had investigations for students that also use tomato seeds. So there are a variety of seeds and plants that have been grown by students or for students.
Okay, let's go here. Is there another one?
My name's Kevin Guerrero, social media, and I'm also a freshman studying aerospace engineering. Now, I know not everyone that studies engineering can work with NASA, but a lot of people work with SpaceX. So what does the future have for SpaceX and NASA working together, like on missions?
One, I think, great example that you'll find interesting is that we've been working to do microsat deployments, both from SpaceX directly as well as from a new capability that's being added on ISS. And so there are university students, engineering students, that are building some of these microsats as university projects, and they are able to use that little bit of extra capacity either on the SpaceX flight or from the ISS to launch those microsats and do studies with them.
Okay, let's go here.
Todd Halverson, Florida Today for Julie. Just to clarify, the 71 hours in a single week was U.S. only?
That was U.S. only, U.S. side only. So the Russian research time was on top of that.
Okay, and do you know how much Russian research time there was that week?
In that particular week, I don't have the sum for that week, but they average about 30 to 35 hours a week as well on their side.
Okay, and do you know what the previous U.S. record was?
You know, we've been bumping that record up gradually. So I think before we hit 71, it was 68, and before we hit 68, it was 63. So we've been basically, as we've ramped up research, we've had these surge days over the last year. So what's happened is we've achieved that overall strategic average that was the goal of the ISS when it was designed, and we see that in some of these amazing days where, they kind of balance out. There's a few days when, right after SpaceX docks, for example, we'll be unloading cargo as fast as we can. And so some of these days with these big surges make up for that and average it out.
And just one more from me. I'm curious as to whether there is any competitiveness between exploration or expedition crews in terms of how much science they can accomplish during their tenure on board.
I would say it's cooperative and collaborative. So crew members help each other. They embrace the research mission. A lot of our experiments need 2 people, maybe one is an operator, sometimes one is a subject, or they need 4 sets of hands to be successful at doing harvesting or something like that. And so they really help each other out, and it's just to get all the research done they can.
Okay, over here on the left.
Amy Gravel with the NASA Social Group. Can you elaborate on the process involved with going from having a scientific question to getting an experiment aboard the space station?
start by splitting. I'll start by splitting your question, because one of the really neat things about the space station is there are two pathways today, and you see that represented on the dais here. So there's one pathway which is to get your research funded through NASA through a competitive peer review cycle that way, competing for NASA mission-driven goals, and those are in research announcements, and that's how Dr. Gilroy, and that's basically the program that Dr. Porterfield's managing. Then the other way is to come through ISS as a national laboratory. In that case, you bring in your own funding, you raise your funding, and then you can enter and do experiments on ISS. And so that's how pharmaceutical companies, educational institutions, and so forth can come through without getting in line for the NASA funding. And having both of those routes to ISS is a real strength of the laboratory. It's how research competes in the real world on the ground, right? You don't have to go through only one agency, only NSF, to get research funded here on Earth. There are a number of different ways you can get your research funded, and that really spurs scientific creativity. You guys want to add to that? Or?
Sure. I would say that in a lot of cases, too, we have, you can be an entrepreneur on the space station these days, too. So if you have the money, if you have the funding and the support to develop your own science, there are very short paths through NanoRacks, CASIS, through the National Laboratory to get on the space station very quickly. In some cases, typically we get researchers on board in about 9 months from the signing of a contract to actual flight. Some cases we've done it as fast as 2 months. I don't like to do it that fast, but 9 months is a typical cycle, 9 to 6 months. And the beauty of the utilization of the U.S. National Lab, too, is that as time goes by, the amount of infrastructure that's on board to cover science experimentation increases. It's a fantastic place nowadays. It's getting better. As I think Julie said earlier, they're flying a flow cytometer. This is a very important piece of research equipment that will help a lot of the biological side of the world. But really, in all aspects of microgravity and space environment research are very well represented now, and we're continuing to build up that infrastructure. So, in the past, when you would fly things on the shuttle, you'd have to basically build your own research hardware that would support your research experiment. And now it's possible in cases even like BRIC, for instance, a researcher can use equipment that they're familiar with on the ground, commercially available equipment that's used in standardized equipment that's used in laboratories. In the case of the protein crystal growth work we're doing, these systems are almost robotic in nature or semi-automated in nature, so we can increase the throughput, increase the number of samples we fly. I like to say it's you're increasing your number of samples, and this is something that it's exploding. It's exploding on the ground, and we're able to take that hardware and use it in the microgravity environment on the National Lab and conduct some fantastic experiments. So I think in the next 2 to 3 years here, you're going to see some really interesting developments coming out of the National Lab side of the space station.
Yes, Simon, you also flew on an expedited schedule. Do you want to talk about your timing to flight?
Yeah, we went through a remarkably quick process. So the classic peer review system tends to just have a lag built into it because it takes time to send out your proposal to other scientists, get them to get feedback, and then of course it's NASA, so then there's paperwork that gets involved after that, and that paperwork for all agencies, going from the decision to be funded to actually the like money to hire someone to begin to do that work, tends to be a very protracted process, and it's just inherent in how the system works. For our experiment on BRIC 17, we went from writing a proposal to having the money appear within the University of Wisconsin system in maybe 4 months, 3 months, something like that, which is a remarkably quick process for us. And to some degree, that kind of changes the game about how you think about doing that kind of research, because, we write the proposal, and then very soon afterwards, we're in a position to get into that ramp-up period where, the flight is coming, there's a deadline, and you can start running for that deadline very, very quickly. So it was a much more painless process than normal.
And if I could, I would add, so a lot of what you heard is the model for CASIS. We're trying to expedite the time from funding. A call goes out to an investigator to the time that you're actually funded to start preparing your payload to actually get your science up. In addition to that increased capability and that shortened time frame, CASIS is also accepting unsolicited proposals. So you don't have to wait for an agency to tell you, we're interested in looking at this particular area of investigation and we have funding available. Write your proposal, we review it. The unsolicited proposal website is open 24/7. So the process is relatively painless for a proposal, and if you have your own funding, if you have identified a person, a company that can help you get your science up to station, that better enables you to put a proposal together and get it in. But if you're coming in off the street, you haven't flown before, CASIS will help you identify an implementation partner that can help your science be done on station. So the goal again is to enable the utilization of the National Lab to its full capability by engaging a broader spectrum of investigators out there, whether they're from universities or companies, pharmaceutical companies, or high schools and middle schools.
Okay, let's go there. Back here to that.
Hi, David Ruck with astronautmovie.com. Can you talk a little bit about where these commercially sponsored research projects sort of fit on a timeline? Is this sort of at the beginning of that? Are we? Has this been going on for a long time? And in more general terms, how do you see the evolution of commercially sponsored research sort of taking place down the road?
So there have been commercially sponsored activities on the space station from the very earliest utilization. Some of the very first experiment on the International Space Station was a protein crystal growth investigation that had commercial collaboration in it. But what we saw in 2005 when Congress designated ISS as a national laboratory, that really opened up the potential. And then once assembly was complete, that's allowed CASIS to really start bringing those commercial and other government agency opportunities to the fore, because we really have, as you've heard, great capacity on the ISS to meet the needs of a large number of users. And so it's really great to see both the more traditional research, if you will, that Marshall's program is selecting, and then the way that they're innovative in terms of open source science. At the same time, you've got commercial R&D having equal access, and it just provides the opportunity for real innovation and for things to advance quite quickly.
And to give you an idea of how fast this is occurring, NanoRacks really started the latter part of 2009. And as of today, we've flown something over 70 experiments to the International Space Station. So it's happening very rapidly. I've been in this business for 22 years. I've never seen this kind of action before. It's a very exciting time.
Okay, down here.
Hi, it's Pete Coons with NASA Social. This is a question for Simon. Regarding the mouse ear plants, do they need to come back, and if so, when? And if they don't need to come back, who will be taking care of them on the station?
So, one of the challenges is for science on the station is if you can't do your analysis on the station, stuff has to come back. And, SpaceX makes that possible now. So our experiment and the co-experiments which are going up with us will go up on SpaceX 2, and they'll come back down, hopefully, on this same Dragon. And then all of our analysis is going to be done with there's a freezer on the station, so we will germinate the plants in space, and then after about 10 days, they get thrown in the freezer, and they'll come back down frozen.
Okay, we'll go over here, and then we'll come over here, and we'll probably have to wrap up.
Okay, hi, Corey Foy with the Social Media Group. One of the questions I had, Mr. Johnson mentioned the the ability to use commercially made and reusable components that are standardized. And with the availability of those and programs like CASIS, is there a queue building up where prioritization is going to need to happen of the mix between commercialized and research projects where we're going to have to figure out, well, not all of them can go up at once. Is that a challenge that you're running into? And if so, how is that going to be addressed from a prioritization perspective?
Well, my hope is to never turn anyone away, right? But I think that prioritization is an important aspect of this, and there will come a time when the capacity is exceeded. We're not there yet, and I'll say too, there's kind of two dynamics happening here. One is we're shrinking. I think the whole community is shrinking the amount of volume, amount of mass that's required to do this science in. And at the same time, the demand is increasing. So they seem to be keeping like kind of a flat level where we can actually accommodate the payloads that are coming in. For instance, I didn't finish my sentence earlier. We've flown 70, but we have about 150 other payloads in the queue right now. And it's really the timing issue with it is just how fast can the experimenters get their experiments in order, ready to go. A lot of times I think the system is actually faster than what the researchers are, which is an odd case for me to see after all these years, but it's refreshing. And yeah, there's a lot of demand. So this is a good time.
But people sort of go in with an assumption that the space station is a fixed finite capacity, and as we fill it up, there'll start being a lot of competition. But actually, as a program, we have amazing flexibility to change out parts, to add more research facilities, to expand those capacities. So every time we see a resource starting to limit our users, our goal is to get the maximum research return for all our users. And so we find ways to expand the system. You know, it's the size of a 4-bedroom house, and so it's really easy to cram in a little extra facility in the corner to meet another user's need. And so that's our goal, is to keep stretching that so that we actually don't ever hit that point where we're turning away outstanding research.
And there's two factors really here that limit the amount of research. One is the up mass for the launch, and the other is crew time. And it's with setting the new record, that's very impressive. But I'm sitting next to Simon. I know he's thinking 70 hours. That's one graduate student for 1 week. But what's really impressive is that 70 or so hours is equivalent to 460 hours worth of research on the ground, because so many of these systems are automated and they're operated on the ground by operators. So that really increases productivity of the station, and you'll see more developments in that respect in the future too.
Okay, Irene.
Irene Klotz with Reuters. The cargo manifest lists a couple of Japanese experiments. I don't know how well you are familiar with all those, Julie, but if you have any information all about the stem cells, what kind of stem cells from what sort of creature, and what's the gist of the experiment?
Yeah, so, you know, as you know probably, stem cells are cells that have not differentiated yet, and they can occur, you know, in any living organism. They haven't figured out what they're going to become as a tissue when they grow up, if you will. In the Japanese stem cells investigation, they're focused on the effect of radiation on some mouse stem cells, and so they're going to fly the cells to orbit. They'll be exposed to the radiation and the microgravity environment for a period of time. Then they'll be brought home, and they'll add those stem cells to an 8-cell mouse embryo. So it's all a mouse-based study, but the mice will all be on the ground.
Okay, one more back at the back and we'll wrap up.
Hi, I'm Christina Russo with Social Media and Public Library of Science. And my question is from Michael Johnson about the protein crystallography project. You mentioned there's 25 different protein crystals or different proteins going in, and I wonder what drives the decision for which proteins to go, and what do you expect for them to happen in microgravity?
Excellent question. Well, actually, the first set this is our maiden flight of this experiment, and the first set that we're flying are standards that have flown in the past. Lysozyme, I think, is one that comes to mind. But what's different in this situation is we're able to gradually change and have multiple samples of concentration gradients, change the concentration across this very long capillary system that we're using, microcapillary system. So, for instance, with the one lysozyme crystal or protein, we'll be able to have 400 different conditions, and we're expecting to see improved crystal growth in the microgravity situation. And what this means is, for other, say, protein crystals, for instance, my favorite that I want to go after in the next few flights will be membrane proteins. Membrane proteins are the fundamental proteins that create cell walls in a cell, and they control every aspect of the cell's metabolism and function. The problem is, terrestrially, these protein crystals are very hard to grow, and most don't grow, in fact. So without being able to grow that crystal, you can't characterize what this protein is, and so the membrane proteins have been quite a mystery for some time now. And what we're hoping to see in the future, we don't have any membrane proteins flying on this particular set, but we want to fly these soon and see what's happening, and we're going to see all sorts of crazy things come out of this. In particular, with the drug research, a lot of times what experimenters will do is combine drugs with the proteins to see how they interact, and you'll get some very specific, say, medications and pharmaceuticals that will come out of this. So that's our potential hope.
Okay, that's going to wrap it up for us. We want to thank all you guys for coming. Coming up next on NASA Television is going to be a heliophysics briefing on the Van Allen probes. That'll begin at 2 p.m. Eastern Time here on NASA Television. We'll be back here at the Kennedy Space Center at 3 p.m. Eastern Time for the pre-launch briefing about tomorrow morning's SpaceX Dragon launch from here at the Kennedy Space Center. And of course, as always, you can log on to the NASA website to find out more about this mission or what's ahead for Expedition 34. That address is www.nasa.gov/station. And as Julie mentioned at the beginning, if you'd like to follow along with the space station science and research, their Twitter handle is ISS_research. So once again, thanks for joining us. We'll see you back here at 3 p.m. Eastern.